All right, good afternoon. It just hit 12 o'clock Central Time um, here in Iowa. Hope everyone is doing well. And if you're here in Iowa, staying warm. I'm Matt Helmers with Iowa State University, ISU Extension and Outreach, and Department of Ag and Biosystems Engineering. Really happy to be able to moderate the webinar today. And for me as an ag engineer, I'm going to learn about something different. I know very little about nematodes um, and what beneficial nematodes can tell us about soil health. So we're really fortunate to have Dr. Christine Sprunger, that's Associate Professor of Soil Health at Michigan State University, to talk to us today. Um, Christine, the virtual floor is all yours. As always, if folks have questions or comments, please type those into the chat box and we'll get those to Dr. Sprunger at the end of the talk. With that, it's all yours.
Excellent. Well, thank you all for joining us today. And I'm excited to talk to you all about soil health and nematode communities. So I always like to kind of start out by just discussing why there's been this heightened interest in soil health. And the first thing I like to do is just give a definition of soil health just to make sure that we're all on the same page. So there are several definitions, but I think the most common one is that soil health is the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. But I think what's more important beyond the definition is really thinking about how we quantify soil health. And I think what makes soil health really unique relative to, other kind of more traditional soil testing is that it doesn't just consider the chemical processes in the soil. It really thinks about the biology of the soil and the physical components of soil. And why we care about soil health is all of these important ecosystem outcomes that are presumably associated with improved soil health. So some of the key kind of major ecosystem services or benefits associated with soil health are often, greater biological diversity, more enhanced agronomic performance, and better crop productivity. There's also thought that with improved soil health, we'll have better water benefits, not only from water quality, but better regulation of water quantity. And then, of course, there's always a conversation around carbon storage, organic matter as being really a core principle of improved soil health. And I think why soil health is important at local and regional scales really has to do with what farmers have been telling me for the past decade. Farmers want more information from current soil testing and the testing that they get from largely commercial farms. The traditional soil testing, again, is largely focused on chemical indicators. But again, if we think back to that definition of soil health, it's really important to think about the living aspects in our soils, and that's where I think soil health has an advantage. There's also enthusiasm for soil health indicators that are more sensitive to management. So some of the soil health indicators, in addition to nematodes that I'll talk about today, we've seen are way more responsive to management. Sometimes when we think about soil organic matter, that can take up to 10 years to see differences across management, but some of these soil health indicators are much more responsive, and that can be really helpful if you're thinking about incorporating a different management practice, or you're wanting to see how a new management practice that that's been implemented, how that ultimately is influencing your soils. And I think there's also this pressure to improve soil health and retain nutrients in the soil, just because we know that there's such a connection between what we do on our agricultural landscapes and how that influences larger bodies of water and other environmental issues. But given that soil health is still a relatively new field, I'd say that it's really taken off in the last 10 to 15 years. There's still some major knowledge gaps within the field of soil health. The first thing that we have to consider is that because it considers both chemical, biological, and physical indicators, there are literally hundreds of different indicators that could be considered to measure soil health. And that can get really overwhelming when we kind of think about like, well, what does the story of all these indicators mean? What does that mean in terms of my soil health outcomes, various benefits that I want to see on my farm? And shockingly, one of the things that we've noticed is really missing in the current soil health framework is the direct measurement of soil organisms. And again, I really credit farmers in Michigan who really encouraged me and pushed me to consider like which organisms could be really beneficial to consider in the context of soil health. And so I really, again, Michigan farmers really pushed me to think more about nematodes and how to incorporate nematodes into the soil health framework. So I'll kind of introduce the idea of nematology in the next few slides. So why nematodes and why are they important indicators of overall soil health? And before I get to that, I think it's just important to clear up what nematodes are. So they are microscopic roundworms, and they are the most abundant form of multicellular animals on Earth. If you think about it, actually 4 out of every 5 animals on Earth are nematodes. And they're found in both terrestrial and aquatic habitats. And I think a big misconception is we often think about nematodes, especially in the agricultural space, as being pests, but the majority of nematodes are actually really beneficial for overall soil health. And I'll talk about the different types of nematodes next. So, if you're not familiar with the soil food web, this is a really great depiction of all the different organisms that exist in the soil and how they're interacting with one another. And you can already see that nematodes are right in the middle of the soil food web, which means they have critical impacts on what's happening kind of downstream in the soil food web in terms of microbes, and then upstream in terms of arthropods. And so nematodes are specialists, and they span different trophic levels, and we'll talk about that in a little bit. But because they're specialists, there are certain nematodes that only feed on bacteria. We call them bacteriivores. And then there are other nematodes that are fungivores. That means they only feed on fungi. And then, of course, there are the plant parasitic nematodes. Those are the pests, and those feed on plant roots. And then there are actually nematodes that feed on each other, and we call them predators. And based on the ratio of these different types of nematodes, we can actually learn a lot about how nutrients are cycling and how our overall soil food web is looking in terms of health and complexity. So one way that we can start to link nematode communities to soil health outcomes and other kind of ecosystem functions that we really care about is placing nematodes on an R to K continuum. So if you all think back to your like high school biology class, you were probably introduced to different organisms and them being kind of binned in as an R strategist versus a K strategist. So we think of R strategists probably as like mice, K strategists as like lions, for example. That's often like the span that we think of. But that same span exists within nematode communities. So bacteriivores are considered R strategists because they're very plentiful. They reproduce quickly. They have a short lifespan. And when they contribute heavily to the nitrogen cycle. So I'll explain that more in a bit. But then we have fungivores and even other types of bacteriivores that are kind of in the middle. They have slower reproduction. They don't produce as much. And then the predators are really like truly K strategists. They have really long gestation periods. They don't produce as much. They're really, really sensitive to any type of disturbance. So they're often the first to disappear with any environmental stress. And so because of their different life histories, nematodes can tell us again a lot about kind of ecosystem outcomes that we really care about. So some really famous nematologists back in the day used kind of that R to K continuum to come up with different nematode indices. And really what they're trying to get at is not only is like the biodiversity and like the count of different nematodes important, but how they exist in ratios and just exist as a community is also critical for understanding kind of ecosystem outcomes as well as soil health. So the first kind of index is a structure or maturity index, and this really gets at your trophic complexity and diversity. So how many types of different nematodes do you have in your soil? And you know, is it just dominated by bacteriivores, or do you have a healthy kind of representation of all of the different types of nematodes? Then we have the enrichment index, and this is really getting at again, nitrogen cycling. So bacteriivores are really important because they actually account for 30% of nitrogen mineralization. That's a factoid that most people have not heard of, but again, this is why nematodes are so critically important, especially those bacteriivores within this kind of nitrogen context. So that enrichment index really gets at, you know, do you have the right bacteriivores present and working in your system? And then we have the channel index. This gets at kind of the fungal, the bacterial decomposition pathways. And again, this can be important when you're thinking about, you know, how quickly might nutrients be cycling. And we can think of like fungal pathways as being slower and bacterial pathways as having a quicker decomposition. And then we have a basal index, and this gets at kind of the amount of disturbance present in your field. So how do we sample and quantify nematodes? This is a picture of my PhD student, Tavisha Martin, and we use these funnels, we call them Bierman funnels, to extract nematodes. And this it's a 3-day process, but nematodes are really attracted to water. So we just take 50 grams of soil, we water the soil daily, and then nematodes really kind of swim to that bottom of the funnel. And then after 3 days, we release the clamp into a beaker, and then we just have the water solution, and there are thousands of nematodes per sample. And then we do have to use microscopy to identify nematodes. And I've been talking a lot about different feeding groups, but we actually go to family, genus, and species for identification, which takes a lot of time. Some of the other kind of soil health indicators that we measure in conjunction with nematode communities is soil respiration. So many of you might be familiar with the Solvita test. This is a very similar kind of indicator that's getting at the same kind of microbial activity. You're wanting to understand CO2 production from your soil, and that often gives you an idea of just overall microbial activity and also the overall size of your soil carbon pool. Then we have this permanganate oxidizable carbon indicator. Some of you may have heard this referred to as active carbon. We've, as a field, have kind of moved away from calling it active carbon because it's actually not as active as we once thought, and it's reflecting a more kind of process pool of carbon. And it turns out it's actually a pretty strong indicator of carbon sequestration and stabilization. And then we have soil protein, which I won't talk as much about today, but it is a new indicator that gets at your organically bound pool of nitrogen. So I want to get into some data. So the first kind of question that I'll ask is, does the long-term maintenance of regenerative agriculture improve soil biodiversity and lead to improved soil health? So I'm going to be presenting data from our WK Kellogg Biological Station long-term ecological research site. So this is Michigan State University's largest field station, and it's located in Southwest Michigan near Kalamazoo. And the long-term ecological research site is funded through the National Science Foundation. It was established in 1989. And one of the great things about this site is that it allows us to compare a wide range of different agroecosystems. So we have a conventionally tilled system, and it follows a corn, soybean, and wheat rotation. Then we have a no-till system, so it has never been tilled since, prior to establishment in the 1980s. Also follows a corn, soy, wheat rotation. Then we have a reduced input system, and this system is managed just like the conventional system, except it receives 1/3 of the inorganic fertilizer, and then there's a cover crop. And so that's how it makes up for that reduction in nitrogen. And then we also have a biologically based system. And so this system doesn't receive any external fertilizers, and it just is looking at what a cover crop can do in terms of overall nutrient cycling. And then we move into our monoculture perennial system. So we have a poplar system, and then we have a switchgrass system. And then we move into our polyculture system. So we have an early successional system. So these are systems that are perennial in nature and have about 13 different species of forbs and grasses. And then we have a mown grassland that has never been tilled. And so, again, this just gives us a great continuum of annual versus perennial systems, and then also different rates of plant diversity. So one of the things that is great about having long-term trials is we can monitor different kind of ecosystem services, different communities over really long-term time periods. And one of the most famous nematologists, Diana Wall, was the first person to measure nematode communities at the KVS LTER, and she sampled them in 1991. And then no one had looked at nematodes, you know, since my lab started getting interested in nematodes again. So we measured in 2021. And so we're able to really have this 30-year kind of comparison to understand how nematodes were shifting through time and across these different agroecosystems. So here's the first set of data. And again, we're asking, how are these different feeding groups shifting over time? So here we have the bacteriivore community. On the x-axis, we have all those different treatments I just went through, and it's comparing 1991 data to 2021 data. And what's surprising is that we actually didn't see that much of a difference within bacteriivores, either across the site or between the 2 years. But probably the most shocking finding that we found was that for fungivores, we see this huge shift where we see a significant increase in fungivore communities in 2021 versus 1991. And why this seems really significant to us is that, but prior to this study, it's really rare to see such significant and substantial increases in fungivore communities. And you can see that, as you go across the gradient, we even see differences, especially in the more perennial and diverse systems. And why this is important is that when you see more fungal communities or a more fungal dominated system, that means that you often have greater soil organic matter cycling, which again, we'll talk about in the next few slides. So it's generally seen as a really positive shift. We also see really substantial differences in the plant parasitic community. We see a substantial decrease in 1991 versus 2021. And I think one of the things that's really driving this is just the act of having a crop rotation, in the conventional system that I think has led to a decrease in plant parasitic nematodes. And then you can just see the really, really low levels in those more perennial polyculture systems. So next, let's talk about some of these soil carbon indicators and what we found. So I know it's a lot to look at, so I'm kind of going to highlight using these red circles. So one thing that we see between 1991 and 2021 is a decrease in this POXI value within the conventional system. And then we see a substantial increase in the mown grassland system. So what this tells me is that this conventionally tilled system is losing carbon, and this mown grassland system is increasing carbon accrual. And we kind of see the same trend in the using this other mineralizable carbon or soil respiration value. And so again, what this is showing is that if you can incorporate perenniality or diversity into your rotation, that's when you're going to start to see greater soil carbon accumulation and also just like reduction of disturbance. So this next slide is really getting at kind of the relationship between nematode communities and soil carbon pools and how they're shifting this relationship over a 30-year period. So the way to read these types of figures is when you see points that are really closely, like tightly together, that means that the nematode communities are really closely related. When points are further apart, that means the nematode communities in those systems are much further or are not related or not similar to one another. So, for example, we see that the mown grassland is kind of way off on its own, and then we have like the no-till and conventional system very tightly clustered. And these arrows it's a vector analysis, and what it's showing is how closely these nematode communities are related to these soil carbon pools. We actually didn't see a strong relationship with nematodes and soil carbon in 1991. So let's look at how this graph shifts when we look at what happens 30 years later. So we see a completely different configuration of these communities, which is kind of exciting. For example, that mown grassland and early successional system are really tightly, closely clustered. And then we have some of our other like reduced input system and the biologically based system and the poplar system that are closely related, where we have the conventional system that's way off on its own. And so what this is showing me is that after 30 years, of implementing more regenerative practices, those nematode communities are reflective of more so what's happening in the mown grassland system versus what's happening in the conventional system. And then that both of these soil carbon values are pointing towards these more kind of perennial and diverse systems. That's where we see a significant relationship between nematodes and soil carbon pools. So just to summarize this section, the long-term maintenance of regenerative agriculture is critical for soil biodiversity and soil carbon. Nematode communities become more fungal dominated after 30 years, especially in systems with higher plant diversity and perenniality. And we did see shifts in soil carbon pools, which could be responsible for overall healthier soil food webs. So in the last few minutes that I have here, I just want to give a couple other examples of how we can use nematodes to tell us more about ecosystem functioning and overall soil health. So at the beginning of the talk, I mentioned briefly that we had these different indices. So we had the structure index that tells us about overall biodiversity of the nematode community, and then we have that enrichment index that tells us about nitrogen cycling. And when we plot these types of indices, we can actually get more information on kind of carbon to nitrogen ratios, whether a system might be bacterial versus fungal dominated, and whether a system is like disturbed, degraded, matured, or maturing. And so we'll walk through some data that we have from a different project where we're comparing a perennial system to an annual system managed with manure, poultry manure, and then inorganic fertilizer. And so what we have here is we have this perennial intermediate wheatgrass in blue, and then we have an annual wheat system in orange. And then the squares are the organic poultry manure. And then the circles are the inorganic fertilizers. And so what do we notice right off the bat? The annual inorganic nitrogen system is in this degraded quadrant, whereas the annual squares are in and circles are in on this kind of other side. We have a maturing or matured system, and then both of the perennial systems are in that kind of maturing system as well. So the main takeaway here is that either having perenniality or an organic source of nitrogen is making these soil food webs healthier and more kind of maturing and more resilient. You're in the quadrant that you want to be in. I have a couple other kind of like commonly asked questions. What's the connection between soil health and crop yield? This is just like one tiny graph that shows, this was a master's student who found a negative correlation between the number of plant parasitic nematodes and above ground biomass. That's not news to anyone. But then in that same kind of system, she found a significant relationship between bacteriivores and above ground biomass. And so that's just like, it's one small kind of indication of the relationship between these nematode communities and crop productivity, which is just a common question that we get. And then the last thing that I want to end with really is what are the pros and cons of using nematodes as overall indicators of soil health? So the pros are that these are easy to extract. We can quantify absolute abundance, which you can't do with microbial communities. And we can quantify non-relic soil food web. So all this means is like we can really zero in on the nematode community, and it gives us really a picture of the overall soil food web. And then the cons are that it's labor intensive. It's not high throughput. So even though microscopy is really cheap, we always have to add in labor, which is not cheap. And so it ends up still being expensive to do. There's also a really training intensive process. It takes a lot of skill to be able to identify these nematodes. And then lastly, we're still like working out like varying methodologies, but the field is really advancing as we speak. So with that, I'd just like to acknowledge my lab. They're the ones doing the hard work in the field and laboratory, and my funding sources. And at this point, I'll turn it over to Matt, and also very excited to answer any questions that you might have. Thank you.
All right, really, really informative and interesting information. And we have some questions that have come in we'll get to in a second. So if you are on and want to earn a CCA CEU, please email Elena Whitaker, ALENAW at iastate.edu by 5 o'clock today. Please include your name, the name you entered to watch the webinar, and your CCA number. Again, ALENAW at iastate.edu. Okay, next slide. If you watch this webinar live and are willing to fill out a voluntary demographic survey, we'd greatly appreciate it. Only one survey response is needed for all 2025 webinars. So if you've done that, thank you very much. And if you're willing, we greatly appreciate it. This is voluntary to gather information for USDA funded programs. Okay. And then we're going to have Michelle Balmer next week with Michelle is with the Iowa Department of Natural Resources, and she's going to talk about from the top of the watershed to the bottom of the lake restoration in Iowa. So a little different topic than we have today, but super important. And then I think we might have one last slide. Excuse me, you can find today's webinar and all previous webinars in the ILF webinar archives. That's on our website, or you can go to YouTube, Iowa Learning Farms slash webinars. There's a playlist there. So go search for your favorite topic. All right, Dr. Sprunger, we have um, questions here. We'll start with sorry. Is there a range of size or magnification needed to see the agricultural nematodes?
Yeah, it's a really good question. So again, we can kind of go back to this slide to give us an idea of the size of the nematode
Okay.
communities. The bacteriivores are the smallest, the predators are the largest. And oftentimes, so we have a really fancy microscope that where we have a screen. So this is actually different than the one I showed. So we don't even rely on kind of looking through a microscope lens. We can just use a screen. But that said, there are some nematodes where to identify them, we have to understand kind of the internal organs of what's happening and like if they have one or two gonads and like how long their tail is. And so sometimes we have to use uh, we put them on a slide and then we have to use oil to make sure that we get the right identification to make sure that we're really seeing clearly what we need to see. So yes, different sizes that often bins them in different communities.
Okay. Are commercial soil testing labs able to test for nematodes?
So it really depends.
Okay.
Most are not. And you are gonna likely have better luck by going to a university. So some universities have like diagnostic labs. Oftentimes they're focused more on the soybean cyst nematodes and plant parasitic nematodes. But there are like very few labs across the country,
Okay.
unfortunately, at this point that are able to identify beneficial nematodes.
Uh, have you seen information on whether certain cover crops appear to have a greater effect on nematode population than others?
Yeah, there's definitely research out there. One thing that we've just noticed in our work is that having incorporating clovers is
Okay.
really helpful. And again, that's because you're getting that extra nitrogen source that boosts kind of, bacteriivores especially. And then so you'd boost your like enrichment number quite often is associated with any type of like N fixer. But honestly, more research is needed in that space.
Okay. What is the average lifespan of nematodes, or how quickly might nematode communities cycle?
Yeah, another really
Okay.
great question. And I keep going back to this figure because
Okay.
bacteriophores, sometimes they live for like a day or two. Others
Oh,
live for like few weeks or a month. So they have a pretty rapid cycle.
okay. If I don't have the nematodes I want, can I add them for faster results, or do I just have to build the habitat they like and wait for them to show up?
Yeah, again, such a good question and a common question that we get quite a bit. So, in all honesty, patience is going to be your best bet. You want to create the environment that is helpful for bringing in these more predatory nematodes, the more fungal nematodes. And as I showed, that can take time. And it can take incorporating perenniality, it can incorporate taking plant diversity. There are some commercial kind of products that, they'll try to sell you the fact that, oh, if you add this type of nematode, you're going to get the results you want. Honestly, I haven't really seen that play out in a really like strong and effective way. So I think doing the hard work is probably going to be your best bet.
Okay. Oh, here's an interesting question, I think, given how cold it is right now. How are nematodes impacted by temperature? And what
Uh,
might their activity look like in different seasons across the year?
Another really, really great question. So yes, right now it's like we have a ton of snow in here in Michigan. If I went out and took a soil core, we would have a lot of dormant eggs, and
Okay.
we wouldn't have a lot of activity. But what we see is, once the thaw starts to happen, we have a lot of activity, a lot of juveniles because they've just been hatched, and juveniles are really, we can't fully identify them. So we count them as our, towards our total abundance. But then when we kind of get through the growing season, we start to see more and more adults, and that kind of cycle really continues through the growing season.
There was a question on the treatment that was the grassland mow system. Kind of how often was that mowed and to what height?
Yeah, so we mow it 1 to 2 times a year, and we usually leave about 6 inches on the ground.
Okay. So here, kind of a interesting one. I don't know if looked at it all, but somebody's asking about the interface between the prairie strips and crops. So in Iowa, and I know KBS may have some prairie strips. Can incorporating those perennials in the form of a prairie strip help increase beneficial nematodes out in the crop field?
Such a good question. So if
Okay.
you look closely at this picture, you can see
Oh yeah.
strips that we've incorporated at KBS. And actually I have another PhD student that's asking that very question.
Okay.
How do the implementation of prairie strips influence nematodes? And so what we do is we'll measure like right in the middle of the strip and then 1 meter out, 5 meters out, and then 20 meters out. So we're really trying to understand like the proximity of the prairie strip and also like the nematodes in the row crop space. And some early results that I can share is that the prairie strip does actually improve the nematode community. And we actually see these indicators shifting where we have greater overall structure indices in the prairie strip and 1 meter out. And we also see an increase in enrichment. And so, yes, prairie strips appear to have
Okay.
a beneficial impact on nematodes.
Well, somebody brought it up, and I was going to too, that we need to plant the seed, that
Yeah,
when that work is finished, get the graduate student to come on and give a webinar for sure.
absolutely. Yeah.
Okay. Yeah. Um, Here's one. How does tillage versus no-till affect the nematodes?
Yeah. So this is a really interesting, I think, finding from our work.
Oh yeah.
So in the literature, we generally see that tillage negatively impacts nematode communities. And we generally see that too. We have, you know, difference, like kind of even differences really in the bacteriivore communities. One thing that was really shocking to us is that we actually see the lowest number of fungivores in the no-till system. And one reason why we think that is, and we also see that the no-till system is kind of off on its
Yeah.
own right here. And we actually think that it could be the pesticides and herbicides that are used in no-till systems at a higher rate than our conventional system, simply because we can't control for like herbicides. We are not using or we are not tilling, so we have to use more herbicides. And so we think that maybe added external inputs might be negatively impacting some of these beneficial nematodes. So again, there are always trade-offs with these agroecosystems. Like obviously, we would prefer no-till for so many other soil health benefits, but any kind of extra added internal inputs could also be influencing your communities.
There's somebody, you may not have this information, but maybe and maybe even somebody else on here does. Person indicated that there is anecdotal evidence of reductions in soybean cyst nematode populations and their damage with soil health management practices. They're wondering if there's any published experimental data or sources out there.
Yeah, one thing that's really frustrating with the soil health world and the nematology world is we often are working in our own camps.
Okay.
And so, for example, I'm not a soybean cyst nematode, so
Yeah.
I don't have that data. But there is anecdotal evidence that implementing all of these different regenerative egg practices should boost your overall soil health and decrease these soybean cyst nematodes. But honestly, there isn't that great of like published evidence. So
Okay.
still
I,
anecdotal.
I know Dr. Greg Tilko was on earlier. I don't know if he has anything in particular on that could type it in the chat box. But um, okay, here's one that this question was, I'm curious about the soil depth for soil C measurements, what that was. And oops, just a second. It jumped on me. Sorry, pardon me. Also, regarding the decreases in the relatively active C pool decrease, is that possible that the changes of the active C pools are related to the redistribution among different soil carbon fractions along the soil profile?
That's a great question. So here we're just measuring the top 10 centimeters. And our colleagues, Bill Robertson and Caro Cordova, who I think Caro did either a postdoc or PhD at Iowa State. She's now a professor at Nebraska. Um, they just published a paper showing these soil carbon pools after 30 years down to 1 meter. So I highly suggest that you check that work out.
Okay.
Maybe we can pop it in the chat or send it out afterwards.
Okay.
But really what it finds is that along the same continuum, you see a decrease in soil health or soil carbon in the conventional system, and then you slowly see accrual as you go through the gradient.
Uh, are there any unintended consequences of using cover crops, such as increased populations of non-target pests, for example, slugs, that might also affect nematode dynamics?
Yeah, it's another great question, one that I don't have data for. So here at KBS, we just started a new long-term agroecosystems research trial. So that's part of the LTAR network funded through USDA, and that was established in 2022. And we had a major slug issue this year in our more kind of regenerative agriculture systems. And that's counter to what we're seeing with the nematode community, where we're seeing positive kind of increases in the nematode communities. And so I don't really know what the relationship is between slugs and nematode communities. I think our slug issue is largely because we had a really wet spring. And it's just not really something that we've had to deal with before, but it's also interesting that we found it more in the regenerative practices and not our more conventional systems.
Okay. And you may have brought this up, maybe you said it in the questions, but I've so tried to read the questions and so forth. But what type of cover crops were used, and what was the maturity at termination?
Yeah, so
Yeah.
the cover crops between corn or before corn we have clover, and then before wheat we have rye. And I'm not sure about the size before termination. I would have to look
Okay.
that up.
Okay. That no problem there. Um, so maybe one last one here. This kind of goes back to the question on whether labs are testing for nematodes. And I guess what would you suggest doing? Just testing for the soil health, poxy, respiration, and soil protein?
Yeah, so there's several labs around the country that are accepting soils for soil health testing. So Cornell University certainly has their soil health testing lab. My lab on a very small level is accepting samples.
Okay.
We have finally gotten approval from Michigan State University to accept external soils,
Okay.
and um, we do charge a fee. So we do have the ability to provide these soil
Okay.
health plus nematodes, but I'm not advertising because we're still
You don't want inundated with too
yes,
many, like, yeah.
yes.
Okay.
But I would go with Cornell University for right
Okay.
now.
Okay. All right. I think that's the main questions. So if folks need CCA CEU, please email Alena Whitaker, ALENAW at iastate.edu by 5 o'clock today with your name, the name you entered to watch the webinar, and your CCA number. I'd encourage you to tune in next week to hear from Michelle Balmer from the Iowa DNR about lake restoration activities. And I really want to thank Dr. Christine Sprunger from Michigan State University. This was really, for an ag engineer, this was a super informative webinar. And I think from all the questions that we got and virtual clapping that we've gotten, everybody else thought that as well. If you have any last comments, Dr. Sprunger, the floor is yours.
Well, thank you so much for your attention. And if you do have questions, please feel free to reach out to me. And then you can also check out our website, sprungerlab.com, where we have all of our publications up on our website if you're interested in learning more about our work. Thank you.
Okay.